I'm Bryce Volschow, and my research was the induced oxidative stress in a neuronal cell line using sodium fluoride. My faculty mentor was Dr. LaSalle. So, fluoride. Fluoride is being used as a topical agent in dental practices and helps to prevent or reduce the formation of dental caries and the breakdown of enamel from various bacterial infections. And from these findings that it may reduce such infections and breakdown, that it has then become added to drinking water. And so in the United States and countries all over the world, the drinking water has become fluoridated in hopes to prevent the amount of enamel degradation or decay in the population. But once it's in the drinking water, the fluoride then goes systemically, so throughout the body. Whereas in normal dental practices, the fluoride is just treated on the teeth directly, and then it is wiped clean, wiped off, and it's not ingested at all. So that leads into the concern is what are possible the effects of fluoride once it's in drinking water, and then when we ingest it as fluoride goes systemically throughout the body. So research has been done and has found that the concern comes from high fluoride concentration in the drinking water. So the U.S. Public Health Service recommends that fluoride concentration is zero point 0.7 to 1.2 parts per million. So we're talking about concerns when the fluoride concentration is well over that upper limit that is recommended. So there's another study done that examined 3-month-old babies and then their ingestion of fluoride, and the study found that they were ingesting well over the recommended range, whether their fluoride concentration in the drinking water was higher or if they were just having an increased daily intake of that water. Locations in China, India, and Bengal have fluoride levels well beyond the upper limit. And then more specifically, locations in New Delhi found that that the drinking water had 4.37 parts per million of fluoride. And again, this is almost 4 times the upper limit that was recommended by the U.S. Public Health Service. Experimenters and researchers wanted to then see what are the effects that these high concentrations have on individuals in these areas. So there is a number of connections made is that in locations of high fluoride concentration, they found that there was a higher rate or cases of hypothyroidism. They also find that there was increased cases of dental and skeletal fluorosis. This is just like a brittle. It causes your enamel to become more brittle and weakened, so it's almost just doing the opposite of what fluoride should do. It's causing them to be weaker and easily broken down. There's a connection to osteoporosis, which is the decrease of bone density. So again, it's creating a more brittle substance or bone tissue. There's also connections with enzyme inhibition, and this is kind of crucial because enzymes facilitate all the metabolic and chemical reactions that occur within a cell. So if we inhibit these enzymes, we start to lose function within basic pathways that our cells need to carry out. And also, there's a connection seen that there's impaired cognitive function in individuals where the high fluoride or where water fluoridation has exceeded the recommended limits. So there has been a lot of research on the effects of fluoride on bone and bone remodeling. However, there is an increased demand of research on fluoride's effects on the central nervous system. So fluoride and the brain. Fluoride has the ability to pass through the blood-brain barrier, and the blood-brain barrier is a very selective barrier which only allows very few ions and molecules to pass through, and preventing anything really harmful, anything like bacteria, most viruses or whatever. It can't pass through it because of the blood-brain barrier. However, fluoride does have the ability to pass through it and enter the brain tissue. One study revealed that the intelligence was lowered in areas where the drinking water had high levels of fluoride within it. Another study looking at the rat brains and then under various treatments of sodium fluoride found that there was damage done to the hippocampus within these rat brains. And this is crucial because the hippocampus is known as the region in the brain responsible for this learning and memory storage. So if we start to see damage here, that's where we can begin to speculate why we are having a lower intelligence level in individuals where there are high fluoride concentrations in the drinking water. And another study was shown that fluoride has the ability to induce oxidative stress in cells. And I'll explain what oxidative stress is on this next slide. So oxidative stress occurs when there's an imbalance within the cell. So normally through cellular respiration and other metabolic pathways, there's a normal accumulation of these so-called reactive oxygen species. And these are just molecules they're unstable and they're very reactive, so they can start kind of getting in the way of other pathways, other molecules, and they can disrupt functioning components within the cell. But the cell has an ability to detoxify these reactive molecules, and every cell comes with its own machinery in order to help reduce the accumulation of these reactive oxygen species. However, when oxidative stress begins to occur, when these harmful molecules begin to accumulate and that our own cell's machinery cannot keep up in detoxifying these molecules, so then we start to have, again, an accumulation of these harmful species within the cell. So cells under oxidative stress cannot keep up with ROS accumulation, and it has been seen that as these species accumulate within the cell, we begin to see damage, and it can even lead to cell death. So mechanisms of defense. Again, like I said previously, our cells have a defense mechanism that helps combat these reactive oxygen species. And one of these specific proteins or enzymes that the cell has is superoxide dismutase 1. So this is, again, an enzyme with antioxidant function. And what this does is able to convert superoxide radicals into hydrogen peroxide and molecular oxygen. Superoxide radicals are just one specific form of these reactive oxygen species that accumulate within the cell. And so this superoxide dismutase enzyme is specifically used to help neutralize these superoxide radicals that form. So again, it's helping to diminish the amount of ROS species that are within the cell. So not only does the cell have its own antioxidant defense, but we can actually ingest and take in antioxidants within our diet. And then these can come from various sources. One antioxidant in particular that has been found is epigallocatechin gallate, or where I'll refer to it as EGCG. And EGCG is a polyphenol found in green tea. Polyphenol is just a specific type of molecule that can partake and neutralize these kind of reactive oxygen species. And studies using EGCG have found that it can act as a free radical scavenger, and that it indirectly upregulates genes for the stress response. So again, these EGCG has an antioxidant role, and it's helping the cell handle and neutralize these reactive molecules that have formed and have began to accumulate within the cell. So my experimental overview is that we are treating Neuro2A cells with various concentrations of sodium fluoride. Neuro2A cells are neuroblastoma cells from mice, and that they can differentiate into a hippocampal-like cell line. And the reason I'm using this hippocampus cell line is because this region, again, is responsible for learning and memory. So I want to see how fluoride is affecting this region of the brain as it begins to accumulate in those cells. Also, in my research, I want to see how the co-treatment of EGCG may play a role in helping the cell kind of battle this oxidative stress that might be induced from sodium fluoride treatment. And from the experiment, and in order to obtain results, we're going to observe changes in SOD1 expression. Again, the SOD1 being that superoxide dismutase enzyme responsible for neutralizing the harmful reactive oxygen species. So here on this slide are results of just fluoride treatment alone. And these are results of a procedure called Western blot. And the Western blot procedure allows us to look at protein expression inside of cells through the various treatments of sodium fluoride. So we see on the left, this is a positive control. We aren't using sodium fluoride, but we are treating the cells with colistin. And colistin has been seen previously to induce oxidative stress in Neuro2A. So again, this is a positive control for the experiment. So if we go from the minus colistin to the plus colistin treatment, if we look at this band here, so this dark band here, and then the less dark or the gray band here, those are indicating the SOD levels within the cell. So at no colistin treatment, we have a pretty profound and dark band, and that corresponds to an abundance of SOD1 in those cells. However, once we treat the cells with colistin, we see that this band becomes less dark, or it becomes kind of more gray, and that indicates that colistin is having a effect on the SOD expression, and it's causing it to decrease the amount of SOD within the cell, corresponding with that change in from dark band to a lighter band. Again, it's decreasing the amount of SOD1 protein inside the cells. So if we look at then to the right, the picture shows the various concentrations of fluoride being added to the cells, and again, we're measuring the abundance of SOD1 inside these cells after the sodium fluoride treatment. So we see at the top it goes from 0, 2, 4, and 8 millimolar sodium fluoride concentrations, and we see as we keep increasing the concentration, we see that dark band all the way to the right becomes less profound with each treatment. We see that it becomes a lighter and lighter gray once we reach that 8 millimolar concentration. Again, this is revealing that upon sodium fluoride concentration, or upon sodium fluoride treatment, we are experiencing a decrease in SOD1 within these cells. And that might add to the oxidative stress response, because as SOD1 is decreased, we are losing a pretty important enzyme that allows us to neutralize these reactive oxygen species, but we are losing it. These ROS species are accumulating, and then we might experience then damage, if not cell death. So that's the only the findings I have so far. So hopefully by the next Friday on the presentation day, I will have the results from EGCG treatment, and to see if it has antioxidant effects and it can help alleviate the oxidative stress imposed by sodium fluoride. Also, we are planning on using an ROS assay to visualize or just observe the actual ROS accumulation within inside the cell. And also, we are planning on examining gene expression using a technique called quantitative polymerase chain reaction, or this RT-qPCR. Whereas before, Western blot looks at protein, the protein abundance. We want to then go in and see now how are the actual genes being affected upon sodium fluoride treatment. Here are my references. Thank you so much for coming and listening.